Your guide to membrane protein extraction. Creative Biomart. Membrane proteins have many important cellular functions that are critical to the survival of organisms. They have more than 60% of drug targets, accounting for 20%–30% of total cellular protein. Membrane proteins include intact membrane proteins, transmembrane proteins, and peripheral membrane proteins. Membrane proteins either attach to the lipid bilayer or bind to intact proteins at the periphery of the membrane by hydrophobic, ionic or other non-covalent. The use of surfactants for the extraction of plasma membrane proteins is not efficient, and it is also possible to disrupt protein-protein interactions and alter the topology of membrane proteins. In some downstream applications, such as membrane protein IP, co-IP, enzyme assays, mass spectrometry experiments, the use of surfactant-free methods to separate plasma membrane proteins is far better than the use of surfactants. For many years, the use of surfactant-free methods to extract plasma membrane proteins can be roughly divided into the following three types of methods. A. Traditional sucrose gradient centrifugation (SGU). Traditional sucrose gradient centrifugation was developed in the 1960s and 1970s and is still used by many laboratories for plasma membrane protein extraction. Sucrose gradient centrifugation separates cellular proteins into distinct components and highly concentrated membrane proteins. This traditional method requires a large amount of starting material and the operation process is complicated and time-consuming. B. Aqueous two-phase affinity separation. This method is characterized in that most hydrophilic polymer pairs are incompatible in aqueous solution, producing two coexisting phases that are mutually balanced. This property allows the separation of many bimolecules including membrane proteins. The method is relatively simple and rapid, but the number of starting cells required is relatively high and the yield is relatively low. C. Centrifugal column method. Using the centrifuge column separation technology, the starting material requires only 2*107 cells to separate the cell/tissue into cytoplasm, nucleus, organelle and plasma membrane components. The centrifuge column has a specific pore size and surface properties. When the cells pass through the column, the cell membrane is broken to separate the intact nuclei, and then the plasma membrane protein is separated by differential centrifugation and density centrifugation without using ultra-high-speed centrifugation. It can be applied to other applications such as SDS Page, immunoblotings, ELISA, IP, co-IP, MS, 2D, enzyme activity detection. Collected by Creative Biomart, a protein extraction service provider, including membrane protein extraction. Contact information. Email, contact@creative-biomart.com. Website, www.creativebiomart.net. Address, 45-1 Ramsey Road, Shirley, New York 11967, USA.